Morning, good afternoon, good evening, wherever you are, and welcome to this webinar session on assessing programmatic needs, stakeholder workshop findings on oral cholera vaccines capsules. We'd be happy to know who's with us today, so you can already introduce yourselves in the chat box and tell us your name and where you're from. You can also use the chat box if you have any technical problem, and I'll do my best to help you during the session. If you have any question during this presentation, please use the Q&A box. It's available at the bottom of your Zoom window. You can ask your questions at any point of the session, of the presentation, and our panelists will take them and answer after the presentation during the Q&A session. Finally, this session is recorded. So we'll share with you probably during this week, we will share with you the links to the video as well as the slides by email. I hope you'll enjoy this session, and I'll give the floor to Annalea. Annalea, go ahead.
So welcome, colleagues. Hello to everyone, and thank you for joining us. We're delighted to present to you today the result of a collaboration between the World Health Organization and the International Vaccine Institute to plan and implement country stakeholder workshops designed to assess the programmatic need and the use case for DuoChol oral cholera vaccine capsules. So if we could go to the next slide. So this webinar has been organized for you as part of our commitment to disseminate the findings from these workshops and to share the learnings both on the potential of this innovative oral cholera vaccine, but also on the approach chosen to evaluate the innovation. And so we therefore aim to start out by providing a brief overview of this innovation or this innovative capsule format oral cholera vaccine product, which is called DuoChol. And this will be presented by Julia Lynch from IVI. Then I'll be following that. So I'm Annalea Khan from WHO's Immunization Department's research unit. I'll be providing you an explanation of the innovation framework approach, which is how we engage country stakeholders to gain insights on the utility and uptake likelihood of this innovation from the user perspective. And finally, my WHO colleague, Diana Spasanowska, will go through the results from our workshop before opening to Q&A to hear your thoughts and reactions and questions at the end of our presentation. So over to you, Julia.
Hello, everyone. I'm Julia Lynch. I'm the Cholera Program Director at the International Vaccine Institute. I'm going to introduce you to the vaccine in development that was the focus of this workshop. So first, just a reminder that cholera is a severe dehydrating illness and has an oral fecal route of transmission. It is a non-invasive pathogen, and so the disease is mediated by a protein toxin. There are two structural elements to the toxin. That is the A component, or CTA, cholera toxin A. That's the active component that actually causes diarrhea. And the B portion of the protein is actually non-toxigenic. So aside from the human suffering, which is clear if you've seen a case of cholera, cholera outbreaks themselves cause substantial economic loss and overwhelm public health services. Next. An analysis in 2015 estimated at least 47 countries that are affected by cholera outbreaks, 2.9 million cases and 95,000 deaths per year globally. Much of the world forgets about cholera until there are large-scale outbreaks that get media attention, as happened in Haiti and Yemen over the last few decades. Next. But since 2022, there has been a significant surge in cholera outbreaks, and cholera is currently a grade 3 global emergency. Next. The first cholera vaccine was a killed whole cell bacteria that was injected. That vaccine had modest efficacy, a rather short duration of protection, and was highly reactogenic. And WHO discontinued the recommendation for using it back in the 1970s. That led to the development of the oral vaccines, which used the same material, killed whole cell bacteria, but administered them orally rather than injected. The proof of concept trial that was done to evaluate the effectiveness of these oral vaccines was done in Bangladesh in 1985. And two different oral vaccines were prepared for that trial. The first was the killed whole cell bacteria with cholera toxin B, that's the WC plus RCTB. And the second was just the killed whole cell bacteria alone. And you can see in the table that the efficacy at 6 months following vaccination was higher with the whole cell with the cholera toxin B component at 85%, including all ages, including young children. However, at 3 years, the efficacy was the same in both arms and lower in children under 5 than in adults. Next. This field trial led to two different lines of commercial development of oral cholera vaccines. The first was the whole cell with the cholera toxin B, which became the commercial product DuoChol. Next. DuoChol was WHO pre-qualified, but in terms of delivery in public health contexts of mass vaccination campaigns, it was found to be somewhat complex to deliver and administer because it required the mixture of a buffer solution from a little package. It required taking this vaccine with a buffer solution because the cholera toxin B component is a protein that would be destroyed by the stomach acid. Also, the packaging was a bit cumbersome and led to a rather expensive product for a global health product. It has been a very successful traveler's vaccine and has been shown to also have short-term protection against ETEC diarrhea. And that's because that cholera toxin B protein has essentially a mirror image in the ETEC toxin. And so we find a cross protection. The second line of development was with the whole cell alone. There you see on the bottom. And if you hit next, this led to the development of DuoChol and UV-Col, or all the UV-Col products. This has also been WHO pre-qualified. Simple to deliver and administer. It's a liquid, inexpensive, and this are the vaccines that have been used in the stockpile. Next. So with the availability of an effective and affordable oral cholera vaccine, Gavi approved the funding of a stockpile in 2013. Requests for the vaccine steadily increased, as you can see in the figure on the right. And since the creation of the stockpile, more than 180 million doses of the vaccine have been shipped in support of mass vaccination campaigns, often in response to outbreaks, but also for preventive campaigns. However, currently, the requests for vaccine greatly exceed the available supply, and new suppliers and new products are needed. Next. So considering new products, in 2017, a preferred product characteristics meeting was held and generated these points as desired attributes for next generation cholera vaccines. That included higher efficacy in infants and children under 5 and lower cost. And in 2018, Gavi put out its procurement roadmap, which asked for future products to have improved presentations like the plastic tube or tablets or other country preferences, but also better thermal stability. So things such as a CTC option could be achieved. Next. So this brings us to DuoChol. This is a low-cost, thermostable, whole cell with the cholera toxin B component and presented in an enteric-coated capsule. And that's done because the liquid, the inactivated whole cells, an Inaba and an Ogawa, the two prominent serotypes of cholera, they've been lyophilized together with the cholera toxin B protein, creating a powder. And the powder is put in an enteric-coated capsule and would be administered similar to the current oral cholera vaccines as 2 doses, 2 to 6 weeks apart. The Ogawa and Inaba strains that are used in this formulation are what are called isogenic, that is genetically identical, except for the element responsible for the serotype difference. There's a new way to produce the cholera toxin B, which is also very low cost now. And so this product has all the components of DuoChol, the whole cell, and the cholera toxin B. So it is like DuoChol, but in a capsule. Next. So considering the composition we expect DuoChol to have the same clinical performance that has been seen with the commercial product DuoChol. And that involves higher efficacy, at least for the first 6 to 9 months following immunization, and including in all ages, that is in children under 5. Cross protection against ETEC, again, a moderate cross protection and for 6 to 9 months, but just as has been seen with DuoChol. The cost now we expect to be similar to the current OCV. What's dramatically different is the thermostability. The prototype capsules that have been developed have been held at 40 degrees for 21 months. So we fully expect this product to not require cold storage or cold chain delivery. Also substantially improved is the weight to volume of the product. If you go to next, if you compare or imagine the capsules being able to be supplied in a pill bottle, as an example, with 100 doses in a bottle, and you compare the weight and volume of 100 doses to the picture in the upper right, which is the current liquid presentations where there's 10 doses. So dramatically reduced weight and volume, which will significantly lower product delivery costs for mass vaccinations, also resulting in much less waste removal. Now, there clearly is potentially a disadvantage. That is that young children may not be able to swallow a capsule reliably. And it may, for those individuals who cannot swallow a capsule, require the powder to be redissolved back into a liquid or perhaps sprinkled on food. Next. However, it is to be expected that 85% or more of a population requiring a mass vaccination campaign for a cholera outbreak, 85% of them or more will be able to swallow a capsule. So most product will not have any challenges in being administered. For those who cannot, we are considering various options. That includes dissolving the powder. The capsules open easily, and the powder easily dissolves back into a buffer or a water solution. Sprinkling powder on food for young children, or what we call the mixed delivery scenario, where when planning mass vaccination campaigns, for example, 85% of the product delivered is the capsule, and 15% is delivered in the liquid formulation, so that there is an alternative for those who cannot swallow a capsule. This would still dramatically reduce the logistical challenges and burdens of a mass vaccination campaign. Next. So with the thermostability and the low weight for volume, this would really facilitate delivery to remote populations, including door-to-door delivery of vaccine and self-administration at home for both the first and the second dose, and possibly enable national stockpiles. When we talk about stockpiles currently, what we really mean is vaccine that's held at the manufacturer's cold storage facilities, where it is then shipped, which often takes several weeks to arrive to where a vaccine has been requested because of an outbreak. But without cold storage requirement and with packaging that takes up very little space, a stockpile that is sufficient for at least an initial response to an outbreak could be managed in country and much more rapidly deployed to an outbreak, not taking weeks, but perhaps taking days. When you respond more quickly, actually, fewer doses are required. Next. So where are we with this product development? It's completed its preclinical development. We're currently in the process of producing the GMP material, that is the quality manufactured required for product to be administered to humans in a clinical trial. So that's ongoing right now. And we expect the phase 1 trial to begin in 2025. And then the phase 2 would follow. And in phase 2 is where we would explore the feasibility and acceptability of different administration strategies I described in those children who cannot swallow a capsule. Overall, I think our earliest estimate of availability of this product commercially would be in 2028 or 29. I think that's it. So thank you for listening, and I look forward to the rest of the seminar.
So the basis of our interest to evaluate the DuoChol capsules has been the recognition that when there's poor understanding of the needs and preference of users and stakeholders, you end up with ill-suited products and innovations. So the problem is particularly true for low and middle income countries that have their own unique set of immunization challenges. So our research aim was to understand those challenges and identify which are most relevant to the handling, distribution, and storage of oral cholera vaccines, or OCVs for short, and then determine if they can be potentially addressed by DuoChol's attributes. So part of our assessment included examining the feasibility and acceptability of a product like DuoChol in comparison to the currently used OCV, also discussing implications on cost and policy requirements for either a preventive or reactive campaign context. And the expectation is that these insights gained from country stakeholders can inform the utility profile of DuoChol, shaping its clinical and regulatory pathway, but also helping us understand whether such a product can allow a more rapid and efficient response to cholera outbreaks. So next slide, please. So the methodology we selected to go about this assessment is a workshop-based framework to evaluating innovations that WHO has been developing and refers to as the Vaccine Innovation Framework. And when we adapted this framework specifically to OCV capsules, our objectives were to firstly determine specific relevant cholera immunization barriers, which can potentially be addressed by changing how vaccines are delivered. We then needed to identify which attributes of DuoChol OCV capsules are most critical and why. And we also wanted to understand and validate potential use cases for this innovative intervention and identify the data and evidence that decision makers would eventually need to make a well-informed decision on whether to adopt this product. However, it was made clear that we were neither asking for a commitment to use DuoChol, nor were we making a commitment to supply it. So the discussion remained strictly hypothetical at all times, but nevertheless aiming to familiarize participants with the OCV capsules, their attributes, the development status, and the programmatic potential. So next slide. So we conducted 2 workshops on DuoChol. The first involved a single country, Bangladesh, and the second was a multi-country workshop for cholera-prone countries in East and Southern Africa, which involved Tanzania, Kenya, Ethiopia, and Mozambique. So Bangladesh was selected for the first workshop, which took place in October 2023, because we wanted the perspective of a cholera endemic country with plenty of experience with cholera vaccination and a relatively low case fatality rate. So there is also a strong legacy, in fact, of research and innovation in this country, thanks to the ICDD-RB, which meant they were receptive to this kind of a workshop. And this is why we were able to assemble a wide range of stakeholders from across the immunization system in Bangladesh, which was totaled 59 participants for 2 full days of really extensive deliberations and group work. And the second DuoChol workshop, as I said, took place at the end of February, earlier this year, and drew on the success and lessons learned from our experience, in fact, with another occasion of using this workshop in a multi-country format in the Caribbean sub-region for a different innovation. We used it on that occasion to assess MR maps, but that gave us the experience of doing it in a multi-country format, and that's how we went about doing it in East Africa. And we decided to take a similar approach there for assessing DuoChol, applying pretty much the same criteria for country selection as we use for Bangladesh, however, aiming for countries with a higher case fatality ratio and more diverse scenarios with respect to access to water and sanitation. And because of the travel costs involved for this kind of a workshop, we did have to limit involvement on this occasion from each country to a specific size delegation. But we nevertheless ended up with about 40 participants representing really a nice mix of profiles from policymakers to service delivery professionals. Next slide. So both workshops were underpinned by the same set of principles that define the vaccine innovation framework, namely being country-driven in the sense that there's a strong engagement and ownership from participating countries, structured and systematic to ensure reproducibility and allow for comparisons, evidence-based, both in terms of the foundation of the discussions, but also ensuring the outputs meet the evidence needs of product development. Then being transparent is part of being credible and emphasizing that participating stakeholders as partners, and that really taking a partnership approach in this innovation space, it was part of our key to making sure we had broad representation and the appropriate type of engagement from stakeholders and partners at country level. So next slide. So the innovation framework follows a fairly standardized methodology, which consists of 4 phases or steps, each represented by a different color in this graphic. And we always begin by identifying immunization barriers, typically relying on a situation analysis. And this initial work takes place mostly in advance of the workshop. Then once we're all together in the workshop, we validate the selected list of barriers and move into step 1, which consists of prioritizing the barriers in the context of vulnerable populations. Then step 2 focuses on the innovation itself, understanding its potential impact on the barriers, and we consider then the preferred use cases as part of step 3. And then finally, the 4th step concerns decision criteria and evidence needs, bringing to the forefront the feasibility question. And each of these steps are highly interactive, very dynamic, with participants pulled into a group work of different kinds, sometimes moving around the room to write on flip charts. And there's a lot of, they change in terms of which the groups are, they're involved in and who are the colleagues that they're interacting and discussing with. And generally, the participants are highly engaged in dialogue with lots of very hands-on facilitation by a team of moderators. And the facilitation is really quite key, as that is what ensures all the perspectives across the participants are heard and consensus is reached. And when there is disagreement, we make sure that even that is documented, and the whole process of data collection and recording the dialogue is all documented through an Excel workbook that guides us through the workshop and allows us to carefully document all aspects of the discussion. And so Deanna will now describe each of those 4 steps and the discussion that took place and the findings that came out of those discussions in a little bit more detail, explaining really how we carried out this assessment of DuoChol OCD capsules and what we found out. So over to you, Diana. Thank you.
Thank you so much, Analea. So just before I start, I just want to remind everyone there is a Q&A button where you can add your questions. And once we finish with the presentation, we will be able to engage in a discussion with you through those Q&A. So in the next few slides, we have divided the findings as per the steps that Analea described. So step 1 was to identify what are the key barriers that countries are experiencing now when delivering cholera vaccines. So we're talking about the current vaccine that is available. To identify those, we have conducted a situation analysis using the WHO guidance for conducting a situation analysis of immunization program performance. And this guidance was really useful to help us look through all 7 EPI categories and to use standardized language around what are some potential barriers that countries might experience. This really helped us then to be able to compare these barriers across countries. So once we have conducted that situation analysis, we went into the workshop to discuss the findings. And we wanted to capture what are the subnational differences as well in terms of the barriers that are experienced. So each of the countries was able to identify their own subnational contexts or settings based on some common characteristics of populations that live in those settings. So for example, in Bangladesh, the subnational settings that were identified were urban setting, rural setting. Then as a special group were identified forcibly displaced Myanmar nationals, and then the Chittagong Hill Tracts. In the Afro workshop, each of the countries prioritized the barriers around 3 settings, so urban, rural, and hard-to-reach settings, noting that hard-to-reach might mean different things in different countries based on their national context. The way the prioritization was done was that the stakeholders came together and they discussed the list of barriers, thinking about the specific setting. So, what is the importance of the barrier in the given context or setting? What is the impact of the barrier on coverage and or equity of cholera vaccination? Whether the barrier is amplified during special conditions. So in this particular case, natural disasters such as flooding, for example, is particularly important because it is also relevant to cholera. So we wanted to see whether this barrier gets amplified during these events. And then if the barrier can be addressed through technological innovations. So this last point is really important because it really relates to barriers that could be affected through changes in presentation, for example, or changes in the way that the vaccine is delivered, but they don't necessarily mean that it's just something that could be addressed through programmatic changes. And so using this process, we have narrowed down the list to high priority barriers. And then when compared across the workshops, we have identified some that we would like to share across all 7 EPI categories. So these were the barriers that were most commonly identified across the countries and across the subnational settings. So in terms of program management and financing, countries have said that there is inadequate subnational budgeting. So this can be both in terms of availability and disbursement of funds. And sometimes there is a delay in the availability and disbursement of logistics. So these two challenges make it difficult to start, for example, a cholera vaccination campaign. And sometimes even when they need to respond to an outbreak, there are delays because of these inadequate and unavailable disbursement. In terms of human resource management, often countries have said that there is inadequate supply of health staff, especially health staff that can be fast mobilized for campaigns. There is suboptimal supervision and inadequate training. In terms of vaccine supply quality and logistics, in many countries, there is an inadequate number of cold chain equipment during campaigns. So this doesn't mean that, the cold chain equipment is insufficient at all times, but when there is a campaign and there is a surge of the number of vaccines that need to be stored during this campaign, then it becomes more difficult. There is insufficient equipment for last mile delivery in campaigns. Again, it relates to the fact that during campaigns, there is an increased number of vaccines that really puts pressure on the cold chain that is available in the country. And there are waste management challenges, and then there are challenges with in-country logistics around cost and transportation. In terms of service delivery, their long distance and travel times really make it difficult to deliver the vaccines, especially to hard-to-reach populations. There are special and remote populations that require more outreach efforts and thus more resources to reach them. And in some countries, there are fragile and conflict settings where immunization is disrupted. In terms of immunization coverage and AFI monitoring, often there is a lack of functional AFI surveillance system and mechanisms are not in place for regular data quality and review. In terms of disease surveillance, data is inadequate sometimes or not available in timely manner or not used for action. Challenges in surveillance in hard-to-reach areas were noted. And in terms of demand generation, there is sometimes vaccine hesitancy driven by cultural and or religious beliefs, and there are inadequate social processes to encourage vaccination. Again, here I just want to emphasize that this is a standard language that we have used to make the barriers comparable across the countries, but the nuances might vary between the countries and subnational settings. Once we have identified the short list of barriers that I have shared the highlights of, we went into step 2, which is the identifying the DuoChol attributes to overcome the prioritized barriers to immunization. So in this step, we took the list of high priority barriers, and then we created a list of evaluated attributes. So as you saw, based on the presentation that Julia presented, we already had an idea of what are some potential attributes that DuoChol might have. And we have listed them here, and this list was shared with the participants. Then they were asked to determine what would be the effect of these attributes on the barriers. So it could reduce or eliminate the barrier. It could have an indirect benefit. So for example, it might increase efficiency within the constraints of the barrier by reducing or eliminating other barriers. But at the same time, it could also have a negative effect. It could increase the barrier or maybe impose an indirect challenge where it amplifies or creates new barriers that create programmatic inefficiency that indirectly negatively impact the barrier. In some cases, maybe there is no effect or this is not applicable. So stakeholders were asked to determine what the effect would be, and they could select more than one attribute per barrier and more than one effect per barrier as well. So once we have done the exercise, we have counted how many times an innovation attribute was selected to have an effect on the prioritized barriers. So just few notes on this paper. On this graph, you will see the attributes on the y-axis, the number of barriers linked to the attribute on the x-axis. But please note that there were a different number of barriers prioritized in each of the workshops, so 25 in Afro and 16 in Bangladesh. There were more than one attribute linked to the barrier, more than one effect was assigned to a single attribute, and this graph shows the counts of the frequency of selection. You will see the darker colors showed a reduction of barriers, reduction of the barrier, and then the lighter colors show an indirect benefit. It is evident from the graph that the most commonly selected attributes to have a positive effect on the barriers were the storage temperature, the packaging, the administration, and then the waste disposal, the longer shelf life, the presentation, and finally the indication. And I will go in the next few slides in details through some of these attributes. In terms of negative effects, you can see from this graph that not many of the attributes had a negative effect on the barrier. It was interesting to note that administration was also selected as an attribute that could have an indirect negative effect on the barriers. And this mainly related to the fact that there is a change in the product presentation of a cholera vaccine, which means that there are changes in the administration. And initially, when the vaccine is introduced, there needs to be more health workers training on the new practices. So, for example, in countries where there are challenges around human resource training, this might impose an initial higher challenge. But stakeholders agree that once the training is done, then in the long term, this will not have a negative effect. So we have selected 3 attributes that we want to focus on. The first one is the improved thermostability. So improved thermostability has the potential to reduce or improve efficiency for the largest number of barriers, but the positive impact is not limited only to barriers relating to cold chain and vaccine logistics and supply. One of the identified barriers in Bangladesh, for example, related to the weak implementation of strategic and operational plans, especially for hard-to-reach areas. So stakeholders there argued that thermostability would reduce this barrier, as there is no need for ice packs, for example, and this would simplify the operational plans and their ability to deliver the vaccine in the hard-to-reach population. Also, improved thermostability would help to reduce barriers relating to insufficient subnational budgeting, as the budget for cold chain would not be necessary. And then barriers related to the inadequate number of health staff could potentially be reduced, as one vaccinator could carry many more doses if the vaccine is in a capsule format, and they would administer many more doses, as they will not need to worry for cold chain, for example. And this also reduces barriers relating to long distance for service delivery and difficulties in transport to hard-to-reach populations. These obviously are in the indirect benefit. For example, the vaccinator will still need to travel the distance, but given that they don't need to maintain a cold chain, they don't need to go back to health facility every few hours to replace the ice packs. Instead, they can stay longer in the field carrying more vaccines. In terms of presentations, capsules in a container or a blister pack is a desired characteristics. It reduces barriers related to program management and financing, such as budgeting and distribution to subnational resources. It reduces the need, the barriers related to logistics costs and transport, as it is easier to transport these presentations. It could improve vaccine delivery to special populations, especially in fragile and conflict settings, as it allows for more vaccines to be delivered in a single trip. The presentation, together with thermostability, would reduce the need for cold chain, as I already said, during campaigns. However, as mentioned, it might impose initial challenges, as this is a novel presentation. So, for example, distributing the product might require changes to the current practice. So some of the stakeholders argued that, the immunization program is very well established and they're very used to their ways of working right now. So by changing the vaccine to a capsule format, then this might require changes in the standard operating procedure. But they believe that the reduced volume and weight would still make the distribution overall easier. And I already mentioned that initially there might be an increased need for training for health staff when this vaccine is introduced. The presentation would also simplify the waste disposal post-campaigns. As you saw, one of the prioritized barriers were challenges with waste management. So this presentation could reduce the need for supervision during waste disposal, and then will alleviate the burden during campaigns on already existing waste disposal mechanisms. The final attribute that we're focusing on is swallowability. So as you saw when Julia presented, one of the expected disadvantages was that maybe children under the age of 6 or under the age of 4 would not be able to swallow this capsule. And so we have probed the stakeholders to discuss this further and tell us, what are their perspectives on this potential challenge. They said that the fact that the vaccine requires, first of all, water to be swallowed, for example, at the point of vaccination would not be a significant challenge. So, for example, stakeholders in Bangladesh said that water is rarely available in all households, so this requirement does not pose a challenge. Even when probed, what if the water is not safe to drink at that point of time, given due to a natural disaster, for example, they said that they would expect that by boiling the water, or maybe by purifying the water through other mechanisms, the water would still be available. Stakeholders in the Afro workshop, they said that maybe there would be areas that don't have access to water so easily, but they said that this, by ensuring there is water, would be easier than, for example, hiring cold vans to maintain the cold chain. So they wanted to say that this trade-off is actually beneficial. So if this vaccine is thermostable, even though they need to ensure additional, vans bringing water, it would still be easier to do and less costly than hiring cold vans to maintain a cold chain. The size of the capsule, as we said, is yet to be determined. However, the swallowability of whether children are available to swallow it or not is still unknown. Stakeholders believe this is obstacle that is not a decisive factor on whether to use the vaccine or not, as there are ways to overcome it. They would prefer if the capsule could be dissolved, but they would like to see specific instructions on how to open or to dissolve the vaccine to ensure optimal dose. Sprinkling on food was not something that is desired, as stakeholders believe that this might create expectations from caregivers or beneficiaries themselves that food will be provided as well. And if food needs to be provided, then it becomes logistically very difficult to deliver the vaccine. So once we have identified these attributes and the effect on the barriers, we went into the step to discuss what are the use cases and whether these are desirable use cases for the stakeholders. So we have identified 4 potential use cases. And the first one is whether they would adopt this vaccine for those that are able to swallow a capsule. So, for example, 6 years or older. They said yes, they would be interested to adopt this vaccine. And the advantages of using these vaccines, as already mentioned, is that there is no cold chain required, no smell compared to the existing OCV, lower operational costs, less waste, and easier transport and storage. Use case number 2 was for populations able to swallow a capsule, but there is no access to clean water. So these are for very specific areas that might have challenges around clean water. And here the answers were mixed. So the overall conclusion was maybe. There are some challenges that need to be overcome before being able to use the vaccine for those use cases. So for example, they would use it they see the benefits of using these vaccines for house-to-house strategies where the water will be available. And as already mentioned, for example, in Bangladesh, where clean water is readily available, this use case would still be used. But in other countries such as Ethiopia and Mozambique, in these cases, they would prefer to use UV-Col, for example, instead of DuoChol because of this lack of access to water. The third use case was for those unable to swallow the capsule, so presumed to be less than 6 years. And again, here the answers were mixed. So the conclusion was maybe. Stakeholders said that they would require specific SOPs, so standard operating procedures on how to administer the vaccine, very specifically describing, how to dissolve the vaccine or how to open the vaccine, because they were worried that maybe the powder quantity might be so small and then lost in the process of dissolving it. If the health workers are trained properly, then they would still consider using the vaccine for these populations. If a buffer is required to dissolve the capsule rather than just water, then it would become more challenging to adopt this this use case in some settings, such as rural settings. But this challenge will not be uniform across the country, as for example in urban setting, it might still be relatively easy to ensure the buffer is present. Finally, the use case number 4 was mixed delivery with UV-Col P vaccine. So for children who cannot swallow, they will be given a UV-Col vaccine, and then for the adult population, they would be given a capsule. This is a likely scenario, so stakeholders agreed that this is the preferred use case for children who cannot swallow the capsule. However, there are some challenges, such as difficulties to use 2 separate products, as it requires more micro-planning, training, and community engagement, and there is a potential of programmatic errors and documentation challenges when using 2 different vaccines. To conclude this step, I just want to say that, as Analea mentioned, this was really purely hypothetical discussion, and none of these answers are binding to countries. So them emphasizing that they would adopt this use case or not adopt the use case doesn't mean that they must do so in the future when this product becomes available. So this is just for research purposes. We also explored the ability to self-administer the vaccine. So instead of the vaccine being given by a trained health worker, maybe it is given by a community health worker, or maybe it is given by, it just given to the user and the user takes it themselves. There were some key considerations that the answers would depend on the country and the setting. Some countries were more comfortable adopting this use case than others. Generally, there was a consensus that self-administration of the second dose of the vaccine was accepted across settings. So, for example, if 2 doses are required, the user of the vaccine would come for the first dose, take the dose, and then the second capsule would be given to them, and they can take it later at home without coming to a vaccination point. However, there was a preference for observation while administration. So, for example, one of the suggestions was to have one community health worker that would monitor numerous people and then make a record whether the vaccine was taken or not. The main challenges that were identified was that it is difficult to monitor whether the vaccine was administered. There were some worries that people might not take it, especially in some countries. They were really worried that in settings with low literacy rates or higher sociocultural barriers, so for example, if they're skeptical of the health system, that there might be lack of trust and they might choose not to take the vaccine by themselves. There could be difficulties in tracking AFIs and coverage monitoring, but countries were open, to discussing how these challenges could be overcome by producing, updated SOPs. And then, as we mentioned, some difficulties with access to clean water. The final step was understanding how a decision would be made, whether to use DuoChol in the countries or not. So, for example, if the vaccine becomes available in the stockpile, how would a country decide whether DuoChol should be used in their country or not? So the first one was to discuss who would be involved in the decision-making process. So key stakeholders would include the Ministry of Health, the National Immunization Technical Advisory Groups, partner organizations such as WHO and UNICEF, and also other ministries such as the Ministry of Water and Sanitation or Ministry of Education. Stakeholders believe that the media, the community, and religious leaders would also play an important role in the post-introduction to support vaccine uptake. The criteria for decision-making covers a broad set of topics such as safety, efficacy of the vaccine, WHO pre-qualification, and then whether the country will have any cost for the product, what is the general acceptability of the population, and so on. For evidence generation, evidence needed to influence decision-making includes costing studies, efficacy and effectiveness studies, demonstration pilots. So countries would like to see, how this vaccine would be used in the countries, as well as cost-effectiveness studies. Finally, we discussed more in details the feasibility of the introduction and mixed delivery of DuoChol. So what would it mean to have 2 presentations in the program when the vaccine is given? And there were several policy as well as programmatic considerations that were identified. So to be able to have 2 presentations at the same time, they would need regulatory approval, in some countries, interagency coordination group approval, NITAC approval, and then if there is a need for self-administration, then a change in the self-administration policy is required. And that in terms of programmatic consideration, training of health workers, cold chain requirements assessments, micro-planning, community engagement, and development of communication materials. So I'll stop here and now over to Analea.
So thank you, Diana. So after each workshop, we've invited participants to provide feedback on the workshop and the innovation framework approach to understand their views on how effective and appropriate this methodology is, and to what extent they appreciated being part of these workshops and how we went about soliciting their involvement. And as I mentioned earlier, our workshops are really extremely dynamic and interactive, but also quite intensive. And we really pack up a lot into 2 days. And so we try our best to carefully facilitate these workshops to ensure that all participants feel appropriately appropriately engaged and clear on the process and objectives, but nevertheless, we can never be certain that all participants are satisfied with how we've gone about capturing their views and getting them involved in the workshop process. And so this is why we insist on this survey each time to monitor if we're meeting our workshop objectives, and of course to make sure we can learn what to improve and how we can continue refining what is a fairly flexible process. And so we're pleased to report that these surveys suggest the workshops are very well received. Generally, the response is very enthusiastic, and the workshops seem to be very much appreciated so far, with participants finding the content relevant to their work and the process engaging and inclusive, and the format, quite importantly, also replicable for other innovations. And so we've been really pleased to see this very positive response. So next slide. So our key takeaways, our general conclusions is that our 2 workshops in Bangladesh and in East Africa were a success, being an opportunity that really offered useful results, which we plan to publish in a peer-reviewed journal, besides this current webinar, which is an important part of our dissemination process. And then overall, we believe that DuoChol could address multiple immunization barriers and has the potential to introduce efficiencies which can yield improved cholera vaccine coverage and equity. And the 3 attributes which emerged as especially appealing to country stakeholders, as you heard Diana present, really were the packaging, the presentation, and increased thermal stability. And there are multiple use cases and settings for which DuoChol is suitable, but more consideration really is still needed on how to administer the vaccine among children who cannot swallow it. So next slide, please. So we're reaching the end of our presentation, and we really want to say thank you so much for your attention so far. We will open the floor shortly for questions, but I want to quickly confirm also that you're most welcome to reach out to us by email with any questions in writing concerning be it the workshops, our findings, or OCV DuoChol capsules, and also the innovation framework process. And our contact information is available here on this slide, but you will also be receiving a copy of these slides, and will be able to access this video through the TechNet website. And lastly, I really want to acknowledge the support we received from the regional offices of the Southeast Asian region and the African region of WHO, as well as each of the WHO country offices for our participating countries, with a special mention for our host countries, of course, Bangladesh and Tanzania. And we're so very grateful, especially to the International Vaccine Institute, for the financial commitment that allowed these workshops to take place, which was possible thanks to a grant from Sweden. And last but not least, very big thanks also to our colleagues who run the TechNet platform for allowing us to organize this webinar. So now we'd very much like to hear the reactions and thoughts and address any questions you might have. So please feel free to submit them in the Q&A. And we'll try to address some of those in the time that remains. So thank you very much, and we are looking forward to hearing from you.
Thanks, Analea. So I see one question. So there's a question, whether the list of barriers and challenges will be shared so they could influence other discussions or research around cholera vaccination. So yes, as Analea mentioned, we're planning to publish a peer-reviewed article, so that will be coming up in the next few months, and there we will have the details of the barriers that were identified, so going into more details than what we have presented in this presentation as well.
So I saw that there was a question that started out from Suleiman Ali, but it just says my question is, and I don't see the rest of your question. So if you could perhaps retype it, looks like we missed a part.
So while we're waiting for that question, maybe there is another one. Is there any plan to conduct a post-distribution survey to measure the same attributes afterwards?
So I'm not sure I'm understanding the question correctly. Would that be after the innovation is made available?
I think that's how I'm understanding it. Yes, we got a confirmation. Yes.
So I think that's a better question to put to IVI, who's been supporting some of the product development.
Yes, as I think was mentioned about what would be desirable information for policymakers to decide on use of the vaccine, I think demonstration projects and effectiveness studies and cost effectiveness, and when any new product comes out, those are important components to really understand how the product works in the real world context. So those would very much be imagined that those would need to be done to really, again, understand the true attributes of the product.
Thank you, Julia.
Yeah.
So we have another question. It says, as we're aware, the cold chain is very important when we vaccinate the children. How can we, how can we communicate with the parents this vaccine does not require cold chain? So maybe I can start here. This is a really interesting point that you raised. That was another one of the considerations that stakeholders had around community acceptance. They were saying that given that this is such a different presentation from what we're used to, it will be really important upon introduction of the vaccine to work closely with the community, with the media, with the religious leaders as well, to make sure that there is a community sensitization to explain, what are the benefits of this vaccine, why is it so different, and to make sure that they understand that this is just an innovation of a cholera vaccine. And I don't know, Analea, Julie, if you want to add anything to this.
No, I agree. I agree with what you said. Thank you.
Yeah. Same. That was fine. Any additional questions? Yeah.
I see one in the chat. Why not chewable for the youngest? And this is a really interesting question that actually came up during the workshops as well. And I think Julia will have the answer to this.
Well, it's difficult to answer simply, but certainly one of the goals here is a low-cost product. And this very straightforward creation of a powder and putting it in a capsule results in a low-cost product. Development of something in a chewable form is a completely different kind of chemistry and manufacturing process that will, I think, not be so low cost, and it just hasn't been done. So we would have to start from scratch, and there'd be a lot of development, and someone has to pay for that development. These are not products that make manufacturers a lot of money. So from a commercial perspective, and the desire to have a low-cost product that countries can afford, but one that is still profitable for someone to manufacture, I think that's the space we're trying to meet. And yes, so I think right now there is no prototype for a chewable vaccine for children.
Thanks, Julia. We have another question. So how long we can store the vaccine?
The expected storage or shelf life is 3 years. I think that will be the target of a manufacturer, is to ensure the product is stable for 3 years.
Thank you.
So any additional questions or even reactions or any aspects of the findings that perhaps surprised you, would be pleased to hear from you, or whether you disagree or you have any concerns. Because I realize a lot of the participants in this webinar are, I've noticed through your introductions that we have a lot of participants from countries that would be really good candidates for using this kind of products. I would be keen to hear more. So there's a question also asking, how many doses would children need to take?
The expectation is it would be the same as all the current oral cholera vaccines, which is 2 doses, at least 2 weeks apart.
And that's the same as the current OCV.
Correct.
Yeah. So I don't see any other questions coming in, but I do want to emphasize again what I expressed earlier, which is that we welcome additional dialogue from you. Please do not hesitate to send questions or thoughts to any one of us. You see the email addresses for Julia, Diana, and myself on this last slide, and of course we are hoping that plenty of additional colleagues will be perhaps following this video at a later date, and so questions do remain welcome in the future.
Thanks, Anleya. I see in the chat, Dr. McConan from Ethiopia wrote that a mixed approach, so DuoChol plus UV-Chol, could be a concern from ease of logistics and planning. Age difference of capsule swallowability and need for water is really a concern. So thanks for this comment. I think during the discussion, we really, went into the details of what would this look like. And what I found really interesting, and Anleya and Julia, you can provide your perspective, was this idea that, when we think about innovations, it's not about just, changing the product, the innovation itself, but maybe that there is also a need to be more creative on like how we're evolving the program, how we're, changing the standard operating procedures to make sure that there is an uptake of this innovation. So perhaps this mixed strategy right now seems like a very challenging thing because it is not something that is the standard. But moving forward, and given that, this vaccine, it will take a few years until it becomes available, it will also give us a time to work on development of, the required programmatic changes, the required policy changes that are needed to ensure that this could become a reality. So what I found interesting was that all of the countries were very much open to having those discussions, obviously with no commitment to do so, but they were interested to continue the dialogue rather than, straight up saying, no, this is very difficult, not possible to achieve. So I found that really, really encouraging and really a food for thought to move this discussion forward. We have another question. So it says, I probably missed the comparison, but what is the advantage of capsule versus current liquid form of OCV disregarding thermostability issue? So beyond thermostability differences, what would be the advantages of the capsule?
So if that's to me, besides the thermostability, you have the weight for volume. So you have a much lower weight product so that much more, many more doses can be carried in the same sort of volume. And you have the higher efficacy against cholera, including in young children in the first 6 to 9 months, which is potentially important in an outbreak setting, I would say. So I think those are other significant advantages, both in terms of the clinical performance, but also the logistics beyond the thermostability, and less packaging, so less waste removal needed from a mass vaccination campaign.
And maybe I would add to that certainly the ease of handling. So if you're comparing it to a glass vial, you don't have the concerns about breakage. If you're comparing it, for example, to some current products where there's the need to shake and it just the handling in general is much simpler and more straightforward with a capsule, beyond also the wastage issues related to loss from thermostability and so on. But there's just certainly a huge advantage with ease of handling, also to reduce wastage.
I also found really interesting the stakeholders several times emphasize also the taste of the current vaccine. So they thought, in terms of desirability by the people that are taking the vaccine, that sometimes the taste could be a challenge for uptake. So the capsule wouldn't have that taste. So it was really interesting to see how the stakeholders are identifying these advantages and disadvantages as well. We were looking at both sides. I see a question in the chat. So capsule in younger population request extra cost on clean water. How can we possibly minimize the cost? And then the second part of the same comment is availability and productivity is scheduled for 2020 of the product is scheduled for 2028, 2029. And then it says, is it considered NCP and global roadmap? In Afro workshops, it was planned in 2026, 2027. So I guess the questions around cost for water, as well as the timelines.
Yeah, and Diana, I think you mentioned this in one of the things that came out of the workshop is it would require water for anyone to swallow, but the relative trade-off it's less logistically heavy to bring in a water tanker or for water distribution than it is to have cold chain vans. So where you needed to bring in water, if that was a requirement, it would still be less burdensome than the cold chain. And then with regard to the availability, this is what happens in product development is timelines can get delayed because of technical issues. So we're now projecting a little bit later, a year or so later than we were before.
Thanks, Julia. We have another question. So are there any special advantages or concerns for use in outbreaks or emergencies compared to preventive vaccinations against endemic cholera?
I would say that that's Jan, that's you. Jan, are you asking, you want to hear from participants, not the panelists, you want to hear from I mean, I think that there are definite advantages. I think this is a great outbreak product potentially because of how, again, particularly if it can be used, if there can be national stockpiles, and product can get much more quickly out to, after early identification of an outbreak rather than delaying weeks, because that means more cases and generally the outbreak has expanded. So I think this being able to, store and transport without a heavy logistical burden could enable getting product out much more quickly to an outbreak setting. I would personally see that as a very strong advantage, or a good use case for this product would be in outbreak response.
Thank you. I don't see any other questions. I don't know, maybe if we want to wait a few minutes just to see if there are any new ones coming up.
So I think we will wrap it up there, and just thank you again to everyone for joining the webinar. Thank you for your insightful questions and your interest, and we look forward to continued dialogue on this subject. And as we said, we will be publishing about this, and so hopefully you'll hear an announcement about that in due time. And I also just will take this quick opportunity to thank my co-presenters, Julia and Diana, and also very big thanks once again to Alex Pascuto from TechNet for your support. So thank you, everybody, and we wish you a very good rest of your day.
Thank you so much. Thank you.
Thank you very much. Bye, everyone.
Goodbye.